Hi, and welcome to this little tutorial, which is meant to explain to you, to show you one example to get from fasta sequences to an OTU table using the uparse OTU analysis pipeline, which is available on this website, drive5.com, and which was developed by Robert Edgard. I'm gonna use here only a few options of all the possible steps, which are basically fitted to my type of analysis, and that's the V3, V4 region of the 16S gene, analyzed with Illumina MySeq. So we will first, before you begin, we will review all that you need, all the files that you need before actually beginning, and then we will go through a quick analysis together using just 2 samples. Alright, so before you begin, make sure you have, for the Windows user, make sure you have installed Cygwin, which is a Linux emulation system, so that the command that you type in the command window of Windows that we will use later on will be actually recognized. So you go on this website, or any other website really, on how to install Cygwin. It's all here detailed, how you installed, so I won't go through it again. Just make sure that you know where you install it. So for example, here it's C Cygwin, or sometimes it's a C Cygwin 64, and then you just follow the steps step by step. It's a very quick install. This just need to make a little change in the path variable of Windows at the end. That's what is currently explained on the screen right here, changing the environment variables and then adding the path, which was the path where you installed it. So again, either Cygwin, like in this example here, or Cygwin 64, depending on which version you will be using. And that depends on your system really, whether it's 32 or 64 bits. And so you will just download the setup of Cygwin and then just follow this step. Alright, once you have that, you still need a few samples, so you can browse the website or you can ask them to me. And so what you will need now is, apart from this Cygwin installed software, you will need a few samples and install the usearch program. So to install the usearch program, you just go on this website, fill in the form to ask for the program. The 32-bit version is free, and then you install it very easily. I am using the Windows build, the Windows version, and I downloaded a few databases. So in the end, you should have something like that to start with. So the usearch program, which is an executable, here you can see I have it here, and then you will need two files for the annotations. You don't absolutely need it, but if you do want some kind of idea of the taxonomic affiliation, you will need them, and they are also available on the website, or you can just ask me. And then here are the 2 samples, the 2 mock samples, and they can be either FASTQ or FASTQ.gz if it's compressed. So once you have this, everything is in your usearch file, in your usearch folder, sorry, and then you can get started. So the easiest way is to just go on the bottom left hand corner and just type cmd like command, and that will open the command window. Alright, here we have the command window, and the last thing we need is the actual commands. And so here, these are the commands that we will be copy and paste in the Windows command. So you can see that there are 10 commands here, and we will copy paste each of them one by one. And you can see here you have a quick description of what each command is doing. So for example, this command is actually to decompress the zipped sequences, and it requires all your sequences file and it's creating your sample files.fastq. Okay, and so on. The second command is to actually get all the sequences in one file with sample origin in the header. Along the way, you can always go back to your Windows and see the files that are created and that will be displayed here in the usearch folder. So once you have that, you get the Excel file open, you get this window open, and then you need to go back to the usearch folder. And so here you can see that I'm a little bit not in the usearch folder, so I go out of these folders by pressing cd ... and I do that again. Now I am at the root of C, then I do C usearch. Now if I do directory dir, you see that I have a list, and these files are exactly the same than the ones that I can see here. Alright, so now I just grab and copy paste. So in my case I don't need this line because I already have uncompressed files. So if you have compressed files, please use this line first. We will start from fastq files. Now just do like this. Make sure you copy all the files. So I copy by pressing Ctrl C, then I go here, right click, and then I do paste. Okay, so we are just preparing the files right now. So right now this command was to add a header in each file so that we know exactly, just looking at the sequence identifier, we know exactly where the files come from which sample. Then we use this. This is to actually go from the fastq file to a fasta file. And in my case, since I only have high quality data which are already assembled using Flash, I don't need to do any more quality check, so I just do this. Then I transform a little bit the fasta file. Just a bit of cleaning here. Alright, and now if I look here in my folder, I can see that I have a all.fasta, which is a fasta file with all the sequences from all the samples, and in the header of each sample, of each sequence, I actually have the name of the sample. Now, if you want, you can also use, you can also cut the primers. That's a little bit more difficult than the scope of this tutorial. If you are under Linux, you can just use cut adapt with the sequences which are here. That's the sequences that are fit for our primers, V3, V4 primers. But in this case, we are in a particular case of full coverage. So see, we have full amplicons because we do MySec with two regions, one forward and one reverse, and it's already assembled with full coverage. So we have this, and the adapters were already cut in my case by the service provider, and so trimming is not needed for me. So for this tutorial I won't do any trimming. So moving on, this is an option. You can see that there are no comments here. And now we will start the usearch program. And so the way we do that is we call the usearch with the build that we want, and we first dereplicate. So that means that we don't want to study twice sequences that are exactly the same. So that's a dereplication step. Okay, now I have a new file, as you can see, which is called unique.fasta. Now once that's done, I'm using usearch to actually find the clusters, or OTUs, or if you species if you want. And I'm using a OTU radius of 3, which is some kind of default settings, but you can also put that to 1 if you want finer OTUs. They are the two practices. So right now it's looking for OTUs, and at the same time in this version it's looking for cameras. It's been published in a Nature journal. and it's a Nature paper, and it's more efficient than UKIND, which was the previous version of looking, the previous way of looking, previous and outdated way of looking for chimeras. Okay, now we can go, it's almost done. Of course it's very quick because I only have 2 samples. Right, waiting a little bit more. Now the next step will be, next step is actually generating a taxonomy database. So that's what I will do, using the representative of the OTUs that we just created. So just paste here. Okay, now I'm actually looking, the database is done. I'm using the database to look for the taxonomic affiliation of the representative sequence of each cluster. That's done. And now I actually, once I have the clusters and I have the taxonomic affiliation of each cluster, I still need to go back to my initial FASTA file with all my sequences and look which, for each sample, which sequences are falling in each cluster. And then so that will actually generate my OTU table. And since I already have the taxonomic affiliation for each of these clusters, this way I will get a OTU table which is already annotated. Okay, so see, it's creating an OTU table here. And if I go here, in my Windows area, in the usearch, you have now not only the 2 FASTQ sequences with the actual sequences, not only the all.fasta file, but also a bunch of other intermediate files, and all the way with the OTU representative, all the way to the OTU table. So if I can open that with anything, but with Microsoft Excel it's working fine. So here I have the ID of the sample, and I have the name of my 2 samples, and I have the number of counts of each OTU in each sample. And I also have an extra column here because I did the annotation, the taxonomic affiliation first. I also have an extra column with the taxonomy and a confidence, some kind of confidence of the taxonomic affiliation. Most of the time I'm not using this. What I do is I actually do the statistics that I want to get the relevant OTUs that differ between the treatments or things like that, and then I want more details on this particular OTUs, and the way to do that is to read in the literature. So basically it's a blast on a NCBI gene bank. And so you need a sequence of OTU 502. Let's say you are interested in this particular OTU because it varies between your samples. And so if you want the sequences of OTU 502, you can go back here and then go to OTU rep, which you can open with any software. I recommend Notepad++, which is a small text editor, which is very nice. And here you have the list of the OTUs, and you can go at OTU 502, and then you just copy paste this OTU. Let's do that. Copy paste with do NCBI blast, nucleotide blast. You copy paste your sequence, you select the options that you want, or you just leave everything on the default options and start run. Now it may take a while depending on at what time you start your query. So right now it's searching. and you can see here that according to the taxonomy it's a firmicute, clostridial, and probably a lacnospiraceae. See here, lacnospiraceae, but with a fairly low confidence, so pretty well sure that it's a firmicute. And so if you go back to the results here, you can see that you have a lot of clones here, because that's uncultured, so you don't necessarily have a lot of information here. But you can see that you have some Clostridium thingy not too far away in this particular example. Well, that comes from the rabbits, so there's a lot of uncultured clones in the rabbits, not that many information available. Okay, and so that was it for today. I hope you will be able to now to use usearch to go from fasta files, from one fasta file per sample with the sequences, to an OTU table. Thank you.